Greetings, all conference participants, conference chair, and staff. My name is Charles, and I'll be presenting the research entitled Predicting SARS-CoV-2 Infectious Dissemination Patterns by Removing SNR Compartments from Series Model Equation. I work at the Secretary of State for Education and Sport in Paraná, Brazil, and my research is designed to the predictive methods concerning COVID-19 dissemination, that is, the Series model equation and its variants. The research launches a question that is being very discussed nowadays concerning S and R compartments of Series equation due to the confounding scenario of variables that sustain this model in order to make predictive numerical solutions for COVID-19 dissemination patterns. We can have a glance of how predictive methods are used when we see every day the time series about cumulative daily new cases worldwide, where for each country, region, building, and else distinct patterns of dissemination are formed to each of these samples. So the question is, can we have a global solution for a given series model to every sample and its confounding scenario that sustain this model? If not, and if you check the latest articles since the beginning of pandemics, you might find several types of series model equation with variations of every sort. For example, we have here an image from Centers for Disease Control and Prevention showing the uncertainty of 21 series models. So I launch another question: why these models present uncertainty? Why there are lots of models officially adopted and many others being generated in the scientific literature? One problem we can assume is that the inputs of data are quite different from one model to another one, in a way that distinct scenarios of COVID dissemination can be found for each sample of the analysis. In that point, no homologous data are found as far as the algebraic relation among objects that compose the COVID-19 infection are persistent, with many forms of interaction. Consisting of this information, topological spaces might vary from many methodological assumptions models adopted to explain COVID-19 dissemination. At that point, we don't have a unique series model with global solution for every case that appear to us, and mainly for the S and R compartments of the equation. The successible and removed terms of the equation find itself some limitations that mainly several variables that is can be listed as policies range, individual behavior compliance, and spreading patterns. That is how you can get infected. It could be aerosol, biosol, or ground forms of transmission. For each spreading patterns, we might find dissemination patterns, derivatives of these three main points that can be also extended to these confounding scenarios where we might find the type and duration of health policies adopted by each country, the health infrastructure, population genetics, human variance in terms of biological resistance, that is the symptomatic, pre and asymptomatic cases, epidemic outbreak, infodemics worldwide, the infodemics hitting population in the individual community level, government and media level, globalization aspects, COVID-19 testing availability, reliability, and time of the outcomes, virus mutation, and citizen adherence to the social isolation and social distance present a strong influence in the S and R behavior patterns of the series model equation. These confounding outcomes in each country pose also a challenge to identify exact design and results of SNR patterns for a local and global solution of equations. It was found in this research that COVID-19 disease could be divided in three regions of analysis where each SIR model should be applied specifically. That is the environmental region, where spreading patterns can be found initially with environmental factors influencing the dissemination. The policies region in the middle, where policies find its limitation uh, the policies region where after countries start to adopt some policies, we might find a COVID-19 reduction. And finally, the urban space region where policies finds its limitation to achieve the best result possible to avoid this or seize the disease dissemination. This research did investigated model equations and its specificity in terms of best predictive outcomes for each region presented. However, one point during this pandemic time was noticed, that is how policies do reduce COVID-19 dissemination. And considering that daily new cases of infected people are empirical data that we rely on, the I compartment of the SIR model equation was used in this research in a simple manner. As far as policies present an attractive solution to the non-linear scenario of variables that influence COVID-19 spreading and dissemination patterns, if we get infected people by daily data, we can observe a long time if the number of infected people is being affected or not by the type and duration of policies adopted in a country. For this, the SIR model equation was dismantled and to get the best fitted data for I compartment and cutting off the S and R compartments. The best fitted data using mode statistics to get the desired sample of the population concentrated in the peak of epidemic evolution as shown in this figure and reaching our I compartment formula where the dissemination rate R is equal to the population divided by the time since the outbreak of the epidemic and day of analysis. This formula used in the specific region of COVID-19 dissemination should give us how, in a simple manner, policies are influencing the disease evolution a long time, as well as we can use the same analysis considering the three regions of the pandemics that was said before. Here we have the the numerical results of this method. Results show that if we analyze the local epidemic evolution, trying to get a global solution of the event, or even a local solution specifically, not separating the variables in every region of the country, it worked well in both analyses for 7, 19, and 33 days. The method indicated that which country was growing or reducing in the dissemination patterns of COVID-19, and even with the confounding scenario of infection spreading. White regions indicate no variations in the dissemination patterns. It means policies are expressing outcomes for the bad or good desired results, depending on the policies adopted. And in this time period of analysis, it can be useful to stop a policy or of a country is adopting or adapt it to achieve better results in both ways for the good or bad result. This can count as a country experience, involve exactly the confounding variables that some countries share. Also, the green lines indicate the oscillations are occurring. Sometimes the COVID-19 is spreading too faster or too slower, and all the confounding scenario of variables can be tracked by these mode statistics with a global view of the scenario. And the same way, if a policy was adopted during this period and it could count as a positive information worldwide, it could be indicate a better way of doing a COVID-19 reduction. It all depends of the type of policy, its duration, and how individuals are in compliance with it. Sharing knowledge of this kind can be an advantage in terms of time parameter that also impact concerning the confounded scenario that each country might present. Why this is important? Because we could track the type and duration policies with the patterns formed for each country, and by doing it, adapting the policies to reach the best optimal solution of desired outcomes. By having this predictive method that encompasses policies and a medical indicator, we have a predictive tool. This later part I will be presenting this later part I present was not performed in this research and is a suggestion of future research. To conclude this presentation, we can point some constraints to SIR model in its full compartment use as a global indicator that can fail to achieve a consistent prediction. And in the opposite direction, the method present in this research tries to fulfill layer E microprocess under a given assumed local stability. Time and machine learning mathematical simulations experience fails. Empirical observations and data constraint the lack of persistent homology worldwide. Privacy, Internet of Things that sometimes might not give us real time tracking of the symptoms or people that get infected and else. There is some limitations of this study, that is the time series that this method use around the world. This data is not updated daily in real time exactly. So some delays or some minor errors in the numerical results can be found, but not for long time periods. I thank you very much, everyone, for us watching this video presentation. Any doubts, any questions, please email me or find me in any social media, and I'll be giving you the proper attention. Thank you very much, everyone, and have a good event.